Hi students, today let me talk about digestion absorption of proteins. Very important 5 mark short notes, right? I have divided into 3 or 4 parts. Very simple 3 or 4 parts, right? This is enough for your 5 mark question. Don't worry and don't get confused with after reading the uh, portions from the textbooks. This is enough for your 5 mark questions of question of digestion absorption of protein. Before going to this, we all know that proteases, that is proteolytic enzymes, are hydrolases coming under hydrolase group. They are all actually peptidases. These peptidases, we are having classification, that is exopeptidase and endopeptidase. You all know the meaning of exopeptidase, which acts on the carboxy terminal, and aminopeptidase involved in N terminals, right? Whereas in endopeptidase, trypsin, pepsin, chymotrypsin, like that. So the second point is, this is our all introductory point. Second point is digestion in stomach. There is no digestion in mouth for protein. Digestion in stomach, we are having 3 important points. One is hydrochloric acid HCl. You know what is the role of HCl? It actually produces denaturation for the protein. And second one is maintain the stomach pH at 2 for the digestion absorption of proteins. Second one is renin, that is actually which is present in infants for the purpose of curdling of milk, easy digestion and absorption. Third one is pepsinogen. You know the meaning of zymogen. ZYMOGN. Zymogen means inactivate substances will be converted into activate substances depending upon the situation or wherever the necessary situations, right? So pepsinogen. For this HCl, give the medium for the digestion absorption of pepsinogen, pepsin, like that. So for digestion of stomach, only 3 points: hydrochloric acid, pepsinogen, renin. Okay. Coming to digestion in intestine, second point. In intestine, we are having important 3 points. Okay. The first one is we have pancreatic proteases. Intestine also released intestinal enzymes. Pancreas also released pancreatic enzymes. They are known as pancreatic proteases. Here the medium is 8.0. For the pancreatic juice materials maintains the pH 8.0 for their metabolism, digestion and absorption. We have 2 important pancreatic proteases, that is cholecystokinine and secretin. Okay. In pancreatic juice, we are having trypsin, chymotrypsin, carboxypeptidase and elastases. You all know that chymotrypsinogen only release chymotrypsin. Pepsinogen only release pepsin. Trypsinogen only will be releasing trypsin. Like that, this pancreatic juice will go for the disruption of the amino acid. Here, the protein will be converted into peptides, then go for the dipeptide, then go for the tripeptides. That is pancreatic proteases. Whereas in intestines, again we are having 2 more enzymes, that is aminopeptidases and dipeptidases. They also will be producing free amino acids from peptide, dipeptide, tripeptide, polypeptide, amino acids, protein, everything. Finally, they will be releasing free amino acids. So, for digestion in intestine area also we are having just 3 points. Okay. Now coming to the absorption. In absorption, these are all the introductory points. Okay. The first one is it takes place in small intestine. Usually L amino acid is more absorbed than D amino acid. The transportation is by an ATP dependent, sodium dependent transport carrier or carrier mediated ATP dependent sodium active process, dependent active process. The cycle for the absorption of protein is Meister cycle or gamma glutamyl cycle. Any one question will be there in your university examination, either Meister cycle or gamma glutamyl cycle. This cycle is very specific for only three organs: brain, intestine and kidneys. Brain, intestine and kidneys. For this Meister cycle, these are all the 3 points, important points. First, you should know about the role of glutathione, where it is synthesized from, how many ATPs are utilized for the production of this Meister cycle, continuation of the Meister cycle. Third one is, what is the name involved in this gamma glutamyl transferase. These are all the 3 important points for your Meister cycle to draw the cycle or labeling. Okay. Now we will move on to how to draw the Meister cycle. What are all the products formed and what are all the energy molecules are required, what is the name of the enzyme, how the outside membrane of the amino acid enters into the inside membrane, like that we are going to discuss in the Meister cycle. Okay. Yes. Now, how to draw the Meister cycle for the absorption of amino acids or proteins, right? If your question is 5 mark essay, 5 mark question, short notes question for digestion absorption of protein, means you should draw this diagram. Okay. One name is Meister cycle, another name is gamma glutamyl cycle. Outside the membrane, amino acid is weighted. This amino acid enters into the membrane, combined with the glutathione, GSH, glutathione, and produces two important products. Two important products: cystinyl glycine, gamma glutamyl amino acid. This is the first step. Outside amino acid enters into the membrane, combines with the glutathione, produces two products. One is gamma glutamyl amino acid, another one is cystinyl glycine. For this conversion, the enzyme name is gamma glutamyl transferase. This is the first step. This gamma glutamyl amino acid converted into gamma glutamate utilizes one energy molecule ATP, and this gamma glutamyl amino acid releases one amino acid and then synthesizes gamma glutamate. This is the second step. For this reaction, we require one more ATP. This is the second step. Don't worry about the name of the enzyme. Enzyme is for here only. Right. Now this gamma glutamate is ready for the conversion of gamma glutamyl cysteine with the help of one more molecule of ATP. Okay. So this is second ATP. First ATP, second ATP. Next one is, this gamma glutamyl cysteine is converted into glutathione with the help of another ATP molecule. Here the enzyme name is glutathione synthetase. Okay, because of the ATP utilizing reaction, glutathione synthetase. So the first round of cycle is over. Second, one product is waiting inside the cellule, that is cystinyl glycine, which are dissociated into glycine and cysteine. These two are the very important product. Cysteine produces so many, 7 or 8 products. Glycine, cysteine functions are cysteine are some 10 functions are there. So glutathione combined with amino acid produces gamma glutamyl amino acid with cystinyl glycine. This cystinyl glycine then dissociated into glycine and cysteine. This cysteine combined with Gamma glutamate produces gamma glutamyl cysteine. That is gamma glutamate plus cysteine, gamma glutamate cysteine. Whereas this glycine combined with this gamma glutamyl cysteine produces glutathione. That is why the full name is gamma glutamyl cysteine glutathione glycine. Gamma glutamyl cysteine glycine is the full name of glutathione. So here we are utilizing one more ATP. So totally how many ATPs are here, or how many ATPs are required for the synthesis of the absorption of the protein? 1, 2, 3. That is why in the last class 3 ATPs are required for the absorption of amino acids. Okay. So glutathione is the very important initial component for the absorption of digestion protein. This glutathione, cysteine glycine, second compound important for this pathway, cysteine glycine, because it will be dissociated into cysteine and glycine. Both are very important compound for the amino acid absorption. Okay. So these are all the points for the absorption of proteins or amino acids. Regarding this, we have some extra points. Some molecules, small molecules, macromolecules, large molecules, medium-sized molecules go for endocytosis, pinocytosis, exocytosis. Okay, that is the first area. Apart from this absorption, Meister cycle absorption. Second one is, if there is any problem in the pancreatic juice, they will go for pancreatitis and cystic fibrosis. Or cystic fibrosis and pancreatitis will produce alteration in the pancreatic juice and contents. Third one is Hartnup disease. This is actually not the enzyme defective disease. This is the transportation of amino acid disease, especially in the kidney and intestine. That is why Hartnup disease is very important for your short notes. The last one is, we have two more components, that is substances, which are also involved in absorption of the protein, that is cathepsins and ubiquitin. Cathepsins and ubiquitin are also involved in the absorption of amino acids, right? So after drawing this Meister cycle, don't forget to mention about these words. Each and every word is very important for your digestion and absorption of proteins. Okay.